So to get this started, that's almost a quote of a song somewhere, I want to reintroduce you to our ATP molecule, and I've sort of sketched out in advance. And I just want to remind you, while I'm saying remind you, I'm assuming you've watched certain tutorials in relation to this. I just want to remind you that in the presence of an enzyme called ATPase, what happens is, if I select on here, and I'll just what happens is that we can, in essence, we can release something called adenosine diphosphate, like this. Notice that we've left one P behind. That goes like that. And we can then also have here our little P, as it were. So I'll just copy and paste my little P. Oh, hang on a second. Wrong layer. It was meant to be this layer. Copy and paste my little P. There it is. Copy. It's going to work for me this time and not messed it up. I'm just going to draw it. Let's say that I'm going to draw my little P. I'm going to have ADP plus another P, which I've somehow messed up my layers with. Another P, there it is. Oh my word, James, here we are making a mess of this. Another P, that's there. But also we have the release of energy. And I just want to remind you of that process. And that is happening all the time to release energy from ATP. And of course, that's our only utilizable source of energy. However, we already know that we need to resynthesize this stuff in order to get the energy we need to fulfill ongoing contractions, such as movement, such as running, such as sport, whatever it happens to be. So we have other structures in our cells. We have something which I'm just going to sketch, which is called C, and I'll use this same blue over here, P. Now this thing here is what we are going to know from here forward as creatine phosphate. Now, word of warning, folks, this is also called phosphocreatine, sometimes just called creatine, but we're looking at this structure. And what we can do is we can take creatine phosphate, and at the start of exercise, especially intense exercise, in the presence of an enzyme called creatine kinase, we are going to get the breakdown of creatine phosphate. And what we're going to be left with is we're going to be left with creatine plus a phosphate group, it's meant to be blue, plus a phosphate group, plus we've got the release of energy. Okay, now at the moment these two things are not associated with one another. We have got this energy here has been utilized to power muscular contraction. In essence, this has gone. You know that energy can't be destroyed, but you know that it's been transferred into the kinetic energy stored movement, muscle contraction. But at this point then, we have ADP, we have a phosphagen, and we have some energy knocking about in the sarcoplasm in this cell. So what we're going to do is we're going to bring this down here. That's just a general area. We're going to bring this down here. And what we are going to do is we're going to say that we have got ADP. So there's my adenosine. There's my adenosine diphosphate. Okay, so there's my ADP. We're going to take my P from here and say plus P. We're going to take this energy here and we're going to say plus energy, which we're going to use to bond and bind this phosphagen onto this ADP, and we are going to produce adenosine 3 or triphosphate, 1, 2, 3, all of those, of course, being unified here. In other words, in the presence of creatine phosphate, we've broken down that creatine phosphate, we've released a phosphagen group and energy, and then we've used that phosphagen group with the ADP we had, plus the energy to bind it on, and then we're now back at this start point at which we can start releasing energy again. Now, can I stress to you folks that what happens here, this reaction here, this is an exothermic reaction, okay? But this reaction here, this is an endothermic reaction. So therefore, together, these are what we would refer to as a coupled reaction. A coupled reaction. The products of one reaction are subsequently used or are used in a subsequent reaction. Okay. Now, That's effectively how this system works, and that keeps going and going and keep turning and turning, typically for about 10 seconds of very high intensity exercise. Now what we're going to do is we're going to sort of analyze this system. The first thing I'm going to do is I'm going to ask, what type of reaction is this? Well, we know, first of all, that this is an anaerobic reaction. Notice, at no point have I said the presence of oxygen, but we can also describe it as a coupled reaction. That's a nice description, as we've just said. It's also a coupled reaction, as we've just mentioned. Beyond that, what about the fuel source? What was the fuel source at the start of our system? Well, if we just scroll back up here, we can see that our fuel source was creatine phosphate. So our fuel source, folks, is creatine phosphate, and that is present in the cytoplasm. In this case, I'm referring to a mast cell, or in the cytoplasm of cells generally, and that is there present, waiting to be used. The site of this reaction, well, I've just mentioned it on a couple of occasions. We've got the sarcoplasm. Fantastic. I'm also going to have a look at the active enzyme of this system. We know that the active enzyme was CK, or creatine kinase. Fantastic. Creatine kinase. We're going to carry on analysing this. And again, all of this analysis can be used to describe and explain how the system works, but also can be compared to other systems, for example. What do we have in terms of an energy yield? Well, for every 1 creatine phosphate, we get 1 ATP. Therefore, we've got a yield of 1 to 1. We can also ask the question, are there any byproducts? What are the byproducts of this system? And we're going to say there are none. Now, you should be able to challenge me on that, given what I've said so far, but there's certainly no fatiguing byproducts. But guys, by the way, you might have noticed that there's a little phosphagen over here that we have not done anything with. And also notice that we've got this little creatine that we've not done anything with. And guess what happens in recovery? Those things are bound back together to form creatine phosphate. So we actually even use that stuff up as well. Now, finally, before we move this on to an evaluation, can I stress to you that the intensity of this exercise, as we've said in previous tutorials, would be very high. And the duration, you should know this already, but the duration would be in the region of up to 10 seconds. So I'll actually put less than or equal to 10 seconds. Bit scruffy. Now, before I finish this off, I want to do an evaluation of this system. First of all, what are the positives? Let me do a different green. What are the positives, the advantages to this system? What's good about it? And these are all positives. We've got all reactants present in the cell. Okay, present in the cell. So that means that we've got no delay for example, oxygen. There's no oxygen delay in this system. It produces very high intensity energy release. Okay, high intensity energy. So we can power, explosive movements, power movements, that sort of sprinting, jumping, darting. We've also got no fatiguing byproducts. Okay, so there's nothing about this system which is fatiguing or leftover. Think about lactic acid, which of course we look at in another system. Now, there are also some negatives or disadvantages to this system. What are those negatives or disadvantages? Well, first of all, we've got and you'll find this out when studying other energy systems, a very low energy yield. So we can describe this system as inefficient. And we can also say one of its weaknesses is short duration or temporary. Now, of course, those two sort of considerations come hand in hand. It's very high intensity, it's very short duration, and the energy yield is insufficient. Those things all tied together, as you can imagine. But that there, folks, is our phosphagen system. Oh, just remind yourselves, this is also known as the ATP-PC system. It's also known as the alactacid system. So just look out for those terms in case you come across them. All of them are referring to the phosphagen system we've just covered. Cheers.